Hello dear candidates, welcome to Bio Classes Online. Today we are going to discuss the three important types of the crosses: the test cross, back cross and reciprocal cross. Coming to the first one, that is the test cross. Test cross is the cross between an individual of unknown genotype and a recessive parent. When a cross is made between an individual whose genotype is not known to us with a recessive parent, such a cross is called as a test cross. And there are two possibilities in the test cross. In one of the possibility, that the individual whose genotype is not known to us may be homozygous or it may be heterozygous. If this individual whose genotype is not known to us is homozygous dominant, then all the offsprings formed will show the dominant phenotype, while as if this individual is heterozygous, that is the individual of unknown genotype is heterozygous dominant, then 50% of the offsprings will show dominant phenotype, while as the 50% will show the recessive phenotype. Now let's try to explain this test cross with the help of an example. We know that the purple color of the pea flower is dominant over the white color of the pea flower, and purple color is denoted by the factors capital P, that is the homozygous purple colored pea flower is controlled by the two same types of the factors denoted by the symbols capital P, while as the white color of the pea flower is also controlled by the two same type of the factors which are denoted by the symbol small p. So homozygous purple colored pea flower is denoted by capital P, while as homozygous white colored pea flower is denoted by small p. So purple color is dominant over the white color, and in F1 generation, if we will cross them, we will get the heterozygous, that is capital P. But these plants having the genotype of capital P will be purple colored pea flowers. So the homozygous is denoted by capital P, homozygous purple colored pea flower. Homozygous white pea flowers are denoted by small p, while as heterozygous purple colored pea flowers are denoted by capital P, small p. If we have a plant whose flowers are purple in color and we are asked to determine the genotype of this pea plant having the purple flowers, and as I already mentioned, there are two possibilities. These purple colored flower containing pea plants may be homozygous or may be heterozygous, and if these are crossed with the recessive parent, that is the white colored pea plant having the white colored flowers, so one possibility is that these pea flowers, these purple colored pea flowers will be homozygous dominant, and as you know that, so the gametes will be formed, the same type of the two types of gametes will be formed carrying the capital P, one of the gamete, and another gamete will be carrying the capital P, but the white colored pea flowers will produce the gametes carrying the small p and small p. So if you will cross here, all the plants will have the genotype of capital P, capital P, small p, capital P, small p, and capital P, small p. So in this case, when this plant is homozygous, then all the plants will have the purple colored flowers. But the second case, so 100% purple flowers, 100% flowers, 100% purple flowers will be formed when this purple colored plant is homozygous dominant. So the offsprings all the formed are purple, but no doubt they are hybrid purple, heterozygous purple, but all are formed. So from here we can conclude that this, if the plant is homozygous dominant, then 100% of the offsprings will show the this dominant phenotype, while as if this pea plant having the purple flowers is heterozygous, white pea plants having the white flowers will produce the gametes carrying the small p again. And here, now if this is heterozygous, the two types of the gametes will be formed. One of the type of the gamete will be carrying the capital P, while as another type of the gametes will be carrying the factor small p. So here you can see, when we cross here, the capital P, small p, capital P, small p, small p, small p, small p, small p. So we can say here these are the purple colored, so 50% are purple colored flowers, purple colored flower containing pea plants or offsprings, while as the 50%, these ones, the 50% will be having the white colored flowers. So in this case, the if the plant is heterozygous, then 50% will show the dominant phenotype, and the 50% will show the recessive phenotype. So we can say here that these offsprings, this test cross has made us to know that whether the individual whose genotype is not known to us is homozygous or heterozygous. Or we can say, in other words, you can see here again that if the phenotype of the individual is capital P, that is homozygous dominant, then 100% purple flowers will be formed here, 100% flower, 100%. But if the individual, or if the pea plant having the purple colored flowers is heterozygous, then 50% will be purple colored and 50% will be white flowered. So you can see here again that if you get all 100% purple flowers, then the unknown parent was capital P, or we can say homozygous dominant. But if you get 50% white flower pea flowered offsprings and 50% purple flowered offsprings, then the unknown parent was capital P, small p, that is heterozygous dominant. So we can say this test cross is used to test whether the individual is homozygous pure or heterozygous hybrid. Or we can say, in other words, that this test cross is used to determine or to know the genotype of an individual whose genotype is not known. So this is all about the test cross. Now coming to the second cross, that is the back cross. A back cross is a cross between an F1 hybrid with its one of the two parents. So the cross of F1 hybrid with one of the two parent is called as the back cross. And here again the two possibilities are here. One possibility is, in one possibility, if there is a cross between the F1 hybrid and the dominant parent. In such a cross, all the offsprings will have the dominant phenotype, while as in the second possibility, if there is a cross between the F1 hybrid and a recessive parent, in such a case 50% of the offsprings will have dominant phenotype, while as the 50% of the offsprings will have the recessive phenotype. So we can see here with the help of this example, as we know, the hybrid tall are denoted by capital T, small t, while as the homozygous tall or homozygous dominant tall are denoted by capital T, capital T. So if this hybrid capital are crossed with the dominant parent, you will see here that all the offsprings are tall, that is 100% tall, while as if these F1 hybrids are crossed with the recessive parent, that is the small t, you can see here that 50% are tall while as the 50% are dwarf. So this is about the this is about the back cross. Now coming to what is the use of the back cross. Back cross in plant breeding, back cross is performed a few times in order to increase the traits of that parent. For example, we can say when we cross a crop plant with a wild variety in order to obtain its disease resistance. Back cross is used in plant breeding experiments in order to increase the traits of that parent. For example, a crop plant is crossed with a wild variety in order to obtain its disease resistance. When this cross is made between the crop plant and its wild variety so that to obtain the disease resistance, in this process most of the good traits of the crop plant gets diluted. So this hybrid which is formed between the crop plant and its wild variety is a hybrid and is repeatedly back crossed with its parent, that is the crop plant, in order to transfer the good traits again or back to it. So we can say back cross is used in the plant breeding experiments to increase the traits of that parent. I have given one of the example, when a crop plant is crossed with its wild variety in order to obtain the disease resistance. During this cross, most of the traits get diluted. In order to obtain those traits back to the this hybrid plant, this hybrid plant is repeatedly back crossed to its parents so that these good traits get back transferred to the hybrid. So this is about the back cross. Now the third type of the cross, that is called as the reciprocal cross. Reciprocal cross is the second cross of the same genotype in which the sex of the parental generations are reversed. When we reverse the sex of the parental generation, for example, in the first generation we use the tall plants as homozygous tall plants as male, while as the homozygous dwarf plants as the females. But when we carry out the second cross and we reverse the sex of these parents, that is now tall plants are used as the females and dwarf plants are used as the males. The second cross in which the sex of the parental generation has been reversed is called as the reciprocal cross, and the initial cross is called as the original cross. You can see here the cross capital A, where the capital A is used as a female, is crossed with the small a which is used as a male, is a reciprocal of the cross. This one is the reciprocal of the cross. This is the reciprocal of the cross, where we have initially used the small a as the female and the capital A as the male. So this is all about the reciprocal cross. These reciprocal crosses were performed by the Mendel, and he found the same results when he carried out these reciprocal crosses. So this is all about these three types of the crosses. Subscribe and share bio classes online. Thank you.